Assalamu alaikum everyone. My name is Shahd Alheijani. I'm a fourth year medical student, and inshallah, I'll be giving the second part of the microbiology crash course, which is Pt2 virology. Inshallah, I'll try my best to finish quickly, and we'll go over everything, all the major aspects of virology, inshallah. Starting by characteristics of viruses, moving on to the genome, and then the diseases, and different classification we have for viruses. You can stop me anytime you want, and after every section, we're going to have a checkpoint with some questions related to that section, and we can get started. Bismillah. Starting with the characteristic of viruses. Right. Let's start with the basics. Now, viruses are very, very small in nanometers. So compared to bacteria and human cells, they are measured in nanometers, very small. They're 50 times smaller than bacteria, 500 times smaller than human cells, and they depend on a host cell. They're not living cells, so they depend on a host cell in order to survive, so that they can replicate, they can form the viruses again, and then they leave the cell. Now, in order to leave the cell, they can leave in two ways. They can either bud off the cell, or they can they can destroy the cell by necrosis and then leave the cell. Now, if they bud off the cell, they will take a part of the cell membrane with them. How does that affect the virus? Now, when they take a part of the cell membrane, they will form an envelope. This envelope will have all the the virus receptors and everything attached to that envelope. Meanwhile, if the virus destroys the cell by necrosis, it will just leave the cell without forming an envelope, and all those receptors, the viral receptors, will be attached to the capsid itself. We're going to talk about the parts of the viruses in the coming slides. Don't worry. Now, for the genome, there are different types of genome the virus could have. They could either have DNA, or they could have RNA, or they can have phases. So it can be RNA and DNA, depending on the phase. So a major example of a DNA virus is herpes virus. Major example of an RNA virus is picornavirus. And we have something called retrovirus. Now, retroviruses, they would be originally RNA, but in some of the phases of replication, they would form a DNA intermediate. This DNA could either be incorporated into the cell, the host cell, or it can again form the RNA, and then the virus will replicate and leave the cell again. So a retrovirus can have RNA or DNA, depending on the phase. And the number we can classify them according to the number of strands. So that it could either be single-stranded or double-stranded for both DNA and RNA viruses. Now, this is the structure of the virus. Wait, let me use the pointer. Here. Now, this is the general structure of viruses. In this case, this is an envelope virus. So here you can see this is the envelope. And as I mentioned, the proteins, the receptors, everything will be attached to the envelope in case of enveloped viruses. Here we have something called the tegument. The viral tegument is basically amorphous protein. It will support the envelope. And here we have the viral genome. It could either be DNA or RNA. It could be single-stranded or double-stranded. And it's covered by a capsid. Now this capsid here is inside in case of an enveloped virus. Here you can see a different image. So here you can see the envelope with the glycoprotein spikes or the receptors or everything attached to the envelope. Here you have the matrix supporting the envelope, and here you have the nucleocapsid. So the nucleocapsid is basically here you have the genetic material covered by the capsid. The capsid is made of capsomeres. So those capsomeres will form the capsid. Capsid is covering the genome, and those will form the nucleocapsid. Okay? Now, I know in your lecture, I noticed in the transcription, the doctor mentioned a nucleocapsid and capsid are different things. So he said that you have the genome, you have the nucleocapsid, and then you have the capsid. But according to all the resources I checked, it's basically the nucleic acid core and the capsid forming the nucleocapsid. Okay? Now, these are the structural proteins. Structural proteins are the capsid, nucleocapsid, and all that. Now, we have something called non-structural proteins. Those are the proteins that will help us with the replication. So, for example, you have enzymes like the polymerase enzyme, you have replicase, reverse transcriptase, and those. We're going to talk about them in a bit. But keep in mind that you have structural proteins and non-structural proteins. And this is the envelope. So we said the envelope is from the host membrane. So when the virus wants to leave the cell, in case we said we have either necrosis or we have budding of the cell. So in case it buds, it will take a part of the cell membrane forming the envelope. Why is this significant? What is the difference between enveloped and non-enveloped viruses? Now, in case of enveloped viruses, these can be easily destroyed by, for example, detergents, soap, alcohol, and such stuff, because this envelope is basically from the lipid bilayer, so it is more easier to destroy those. Meanwhile, naked virus, which is it only has the genome covered by the capsid, this is harder to destroy. So, for example, any invasive disease in the GI tract and so on, they're usually naked viruses because they're very hard to destroy, not easily destroyed by the stomach acids and so on. Meanwhile, envelope viruses are easier to destroy generally. Now, this is another diagram. Here you can see the surface proteins in case of a naked virus are attached directly to the capsid. So they can invade, they can attack the cell. Meanwhile, in this case, the surface proteins are attached to the envelope. So when you destroy the envelope, those surface proteins are gone with the envelope, and it's easier to get rid of the infection, basically. Those are different shapes or different appearances for the viruses. So here you can see the icosahedral shape, and the genome is inside, and this is naked because it does not have an envelope. So those spikes are attached directly to the capsid. Here you have a spherical shape. You have the genome inside, covered by the capsid and by the envelope. You can see the envelope will resemble the cell membrane, and it has the spikes attached to the envelope. Meanwhile, this is a different shape. This can be found in Ebola virus, for example. So this is enveloped and filamentous. So it's not spherical, it's not icosahedral, and so on. So this is a different shape. Now, moving on to the basics. Wait, let me check if you have a question. Is my voice not clear? Can you guys hear me?
Yeah, it's clear now, but it was cracking a bit. Now it's clear.
Okay,
Thank
sorry. Do you need me to repeat anything else? Anything before? Okay, good. Yes, what do you need me to repeat? Small recap? Okay. So basically, for viruses, we have the genome inside. Genome could be DNA or RNA, could be single-stranded or double-stranded. This genome is covered by the capsid. Capsid is basically proteins made of capsomeres to cover the genome. And then you can either have the spikes attached to the capsid or to an envelope. The envelope is taken from the host cell. So when the virus, in case it buds from the cell, it will form an envelope. And this envelope will have everything attached to it, the spikes, and the envelope is easier to destroy. So usually the invasive diseases in the like in the intestines and so on, they're usually naked viruses more than envelopes because it's easier to destroy the envelope viruses. This is basically everything we said. We didn't like go into details more than that. Now we're getting into more details in the basics. Now let's talk about DNA viruses. We said one of the major groups of DNA viruses is herpes virus. Let's take this question. Okay. Yeah. So now, one rule for DNA viruses, they're usually double-stranded. We have one exception for the double-stranded rule in DNA viruses, which is parvovirus. So parvovirus is a DNA virus that is single-stranded. Usually DNA viruses are double-stranded, except parvovirus, single-stranded DNA. Rule number two is that they replicate in the nucleus. So you can imagine that DNA viruses, just like human cells have DNA. Replication happens in the nucleus normally. So this is the general rule for DNA viruses. Moving on to the RNA viruses, rule number one is that they're usually single-stranded. Now, the exception for the single-stranded rule in RNA viruses is reovirus. Reovirus is usually double-stranded RNA. So to remember this, you can remember Reo as duo. So they come in duos. So it's double-stranded RNA. Meanwhile, other RNA viruses are single-stranded. Now, when they're single-stranded, this strand could either be a positive strand or a negative strand. تمام? We're going to talk in details about the difference between positive and negative strands, but for now, just remember this. A single strand could either be positive or negative. Rule number two is that they replicate in the cytoplasm. So DNA viruses in the nucleus, RNA viruses in the cytoplasm. An exception to the RNA in the cytoplasm rule is influenza. Influenza is a single-stranded RNA virus that replicates in the nucleus. تمام? Remember that. Now for the appearance, it could either be monopartite, in which all the genome is in one nucleic acid molecule, or multipartite, in which the genome is in many nucleic acid molecules. تمام? طيب. Now, this is a general classification. So the Baltimore classification will divide the viruses into different groups. So for now, all you have to remember is that you have all those groups. Group number 1 is double-stranded DNA. Number 2 is single-stranded DNA, which is a positive strand. 3 is double-stranded RNA, and so on. So in the coming sections, not in this lecture, we will talk in details about every single group, and you need to remember the examples of the viruses in each group. But for now, just remember that you have the first two groups are DNA, double-stranded, then single-stranded, and then the rest are RNA. You have double-stranded RNA, single-stranded RNA. As I mentioned, single strand could either be a positive strand or a negative strand. Now, we said we have some RNA viruses with a DNA like intermediate in their cycle, and this is a retrovirus. And you have a double-stranded DNA with RNA intermediate, which is the hepadnavirus, such as hepatitis B. We're going to talk about everything in detail, bit by bit. Now, we have something called tropism. Tropism is basically the preference of the virus for a specific type of cell. Now, this depends on the receptors on the cell. So in this case, for example, let's take rhinoviruses. Rhinoviruses have tropism for the nasoepithelium. And this is due to the ICAM receptors. So we said tropism depends on the receptors. So they have preference for the nasal epithelium due to the presence of ICAM receptors for rhinoviruses. Meanwhile, moving on to HIV, HIV has tropism for CD4 T cells. تمام? And this is again due to receptors on the surface of the cell. So HIV in CD4 cells, which are T lymphocytes, rhinoviruses in the nasal epithelium because of ICAM, and here you have the COVID virus, which has a preference for this receptor and the nasal epithelium. تمام? So tropism is the preference of the virus for which type of epithelium, which type of cell, depending on the receptors. Now, host range is basically the type of tissues, cells, species it can infect. So it can either be a broad host range, where it can infect many cells, many different species, or it can be narrow, where it can infect one thing only. So for example, you have neurotropic viruses, which are viruses that have preference for the neurons or for the CNS, basically, which are rabies, poliovirus, and herpes simplex virus 1. Those are neurotropic viruses. They prefer the nervous system. Respiratory viruses, they prefer respiratory system, such as influenza, rhinovirus, and coronavirus. Now, liver like viruses with a liver preference, such as hepadnavirus, which causes hepatitis and yellow fever. And finally, genital. Genital is herpes simplex virus 2. So don't confuse between herpes simplex 1 and 2. 1 is basically by direct contact, by the kissing and such, like that. This will have a tropism or like a preference to affect the nervous system. Meanwhile, HSV2 is through sexual contact, so it's genital. HSV1 is through direct contact. HSV2 is through sexual contact. Now you have two terms to remember. You have enzootic and zoonotic. Now, enzootic will be restricted to the natural host or the natural species, so it won't jump from species to species. Meanwhile, you have something called zoonotic infection. In zoonotic infections, they will jump species. So, for example, animals to humans. Some examples for zoonotic infections are, for example, rabies, in which they would jump from like the rabid animals, dogs, and such stuff to humans. You also have dengue fever. So dengue would be transmitted by mosquitoes. So originally it's in mosquitoes and then it will jump to humans. So those are zoonotic infections. تمام؟ طيب. Now moving on to the replication. So replication has 7 stages. Replication for the virus starts by attachment. So the virus first has to attach to the host cell, and then it will penetrate the membrane going into the host cell. It will uncoat to release the genome, and then the genome, there will be replication. So the genome will start replicating, or the proteins will start replicating, and then those 3 stages, penetration, uncoating, and replication, are the eclipse phase. Why? Now, for example, in solar eclipse, lunar eclipse, you would see the sun hiding behind the moon, or the moon hiding behind the sun. So basically, you won't see that thing. In eclipse phase, you won't see the virus because now it's inside, it's uncoated, it's replicating, you won't see an actual virus. So this is the eclipse phase. Meanwhile, in the next stages, you have assembly, maturation, and release. So in those are the productive phase. So now you are actually producing the virus. So the productive phase is the last 3 steps: assembly, maturation, and release. So those are the 7 general steps for replication. Now we'll take them bit by bit. Let's start with attachment, penetration, and coating. Basically, the virus will attach to the receptor. It depends, different receptors for different viruses, as we mentioned earlier. Here, let's take this example. So the virus will attach to the receptor, and now it will be internalized. Now, it could either be internalized by viropexis, in which it will attach to the receptors, it will be taken in by endocytosis, and then goes into the cell, or it will only, the membrane of the virus will fuse with the membrane of the host cell and release its genome. So this is viropexis by endocytosis, and this is membrane fusion, and then the genome is released. Those are 2 different ways to penetrate the cell. Now we'll move on to uncoating. So uncoating is in acidic endosomes. So proteolytic enzymes will break down all the protective layers, and then to release the genome. So those are the first steps. Now we'll move on to replication. So you have nucleic acid and proteins. Now, let's take 2 different examples. We have single-stranded positive RNA and single-stranded negative RNA. Now, what is the difference between positive strand and negative strand? In positive-stranded RNA, this positive strand means we can directly translate this RNA into proteins and start right away. So it will be a faster process. So now the virus, for example, in this case, poliovirus is a positive RNA virus. As soon as the genome goes in, it's a positive RNA.
Someone said if you can repeat the last slide quickly.
The last slide, okay. Yes.
Yeah, someone is asking.
This one. Yes, I just thank you. The attachment, penetration, and uncoating. Okay, so it will attach to the cell. When it attaches to the cell, it will bind to the receptor. Now, to penetrate the cell, it could either penetrate by fusing the membrane fusion. So now the genome is released, as you can see here, or it can be internalized. So by viropexis. Now the host cell will basically swallow the virus, and then inside it will be uncoated, digested by the proteolytic enzymes, and the genome is released. So it's either fusion at the membrane or by viropexis. And then we'll start by replication. So the replication could either take place in the cytoplasm or nucleus. So as we mentioned earlier, DNA is usually in the nucleus, RNA is usually in cytoplasm, except influenza in the nucleus. تمام? Clear? Okay. Now, back to replication. So we said positive stranded RNA, positive single-strand RNA, like poliovirus, it will start translation right away. So it can directly form the proteins, start its own thing, and start with the process. It's one of the first proteins that is formed or that act is RNA-dependent RNA polymerase. Now we will be talking about the specific specific enzymes in the next section. But for now, just remember that you have RNA-dependent RNA polymerase. To understand what this thing does, first thing, RNA polymerase. RNA polymerase means we are making RNA. تمام? RNA-dependent RNA polymerase. RNA-dependent means we are using RNA to make RNA. Okay? So RNA-dependent RNA polymerase is basically the enzyme that will make RNA from an RNA strand. Now, when you think about it, in humans, we're usually making RNA from DNA. So usually what we have in human cells, you have DNA-dependent RNA polymerase. So you make RNA, but from DNA. This means that the virus should have its own RNA-dependent RNA polymerase because the cell can't supply that. The host cell, the human cells, do not have RNA-dependent RNA polymerase. We do not make RNA from RNA. Only viruses do that. So one of the first proteins in this cycle is RNA-dependent RNA polymerase. تمام? Now moving on to single-stranded negative RNA. This is like paramix of viruses. So in negative strands, you cannot use a negative strand to make proteins directly. You need to make a positive strand. So here, you will have polymerase enzyme to make mRNA, the complementary positive strand. So here you have the negative strand. You need to make the positive strand and then use the positive strand to make proteins. and then the proteins will do their own thing, and then they'll be assembled and then release the virus. But you need to remember that positive stranded RNA, they will right away start working, خلاص do their own thing. Negative strand need to go through the whole process to make the complementary positive strand and then start translation and so on. Good? This is for the nucleic acid. Now for the proteins. Proteins, to make proteins, again, you need to translate the mRNA to make proteins. Now, if we have many proteins to make, this is a question. Can you repeat? تمام. طيب. ماشي. You can either have positive single-stranded RNA or negative single-stranded RNA. Now, in positive, a positive strand can be translated directly to form proteins. تمام? So when you translate it directly, خلاص, you form proteins, and then you will form the RNA, form the proteins, assemble the virus, and then release the virus عادي, normally. Meanwhile, if you have a negative strand cannot be used to make the proteins. So you need to make a positive strand. So now you need enzymes to make the positive strand. Positive strand will be used to translated to make proteins, and then you can assemble the virus and release it. That's basically it.
So in positive strand RNA, basically we don't need to transcribe it or like replicate it. We just translate it directly and we produce this proteins for the RNA. And in negative strand, we have to make positive strand so that we can make proteins. Is that
Exactly,
it?
yes. But keep
So-
but keep in mind you still need to make more RNA because you need to make new viruses. So each virus will have its RNA, but you only need to make the positive strand عادي. You don't need to translate to the complementary strand. تمام?
Okay. So we just replicate the RNA and then we just create proteins directly.
We'll have a section about all the replication process in a bit. For now, just remember that the positive strand can be made into proteins. Negative strand needs to make the positive strand in order to be translated. We'll talk about the whole process in detail for every single group in a bit. تمام?
Okay, makes
If
sense.
after that
Thank you.
section you don't understand, just ask me again and I'll repeat. تمام?
Okay, makes sense. Thank you.
You're welcome. Now for the proteins, you can either have a polycistronic mRNA or precursor protein. Now, what is the difference between them? In polycistronic mRNA, you have one mRNA strand encoding many proteins. So when you translate that mRNA, you translate it into different proteins initially. ماشي? You're not forming one protein, you're forming many proteins from one mRNA. So this is a polycistronic mRNA, and this is seen in rabies virus. So rabies virus will have one mRNA making many proteins. Meanwhile, you have a different case here in polioviruses and flaviviruses. In those viruses, you have a precursor protein. So one mRNA strand will make one protein. This is a huge protein. Now, this protein is going to be broken down into different polyproteins, and those proteins will be again broken down and will form either structural proteins, which are the capsid, nucleocapsid, and so on, or the non-structural proteins, which are the polymerase and the enzymes and so on. So polycistronic mRNA, one mRNA, many proteins. Precursor protein, one mRNA, one protein broken down into many proteins. So this is seen in poliovirus, flaviviruses. Meanwhile, polycistronic mRNA is in rabies virus. That's basically it. Good? Now we have different types of proteins. You have immediate early proteins, you have early proteins and late proteins. Immediate early proteins are things that we need right now because the virus entered the cells, we need right now. We need regulatory proteins and to initiate replication. So to start the whole process, in order to make more viruses and start everything, you need to initiate replication. So those are the immediate early proteins. The early proteins are the genome replication things. And then the late proteins, later on, you need the capsid proteins and to assemble the virus and to release the virus. There's nothing much to say about here. خلاص. Immediate early for initiation and regulation, early proteins for replication, late proteins for the capsid and assembly and release. تمام. Now, assembly, maturation, and release. After we made the nucleic acid, we made the proteins, we made everything. Now we need to assemble the virus. We have post-translational modification in case we need to glycosylate the proteins, for example, we need to adjust the proteins, we need to do anything. This is post-translational modification. And then encapsidation. So we take everything, we put it in the capsid, and now the virus is ready. خلاص, we generated all the viruses in this factory. Now we need to release them. To release, it can either be by lysis or budding. Remember we said there's a difference between viruses that live by lysis, viruses that live by budding. So here you can see viruses that live by lysis are the naked viruses. So they will cause the host cell to burst. They release the viruses. خلاص, they're not enveloped. Meanwhile, those that are released by budding, they will be enveloped. So you have the whole protein, you have everything. Now it's released by budding. You have the envelope, and then the spike proteins coming out of the envelope itself. تمام. Here we're done with the first lecture in virology. Any questions before the test?
Yeah, can I just
Yes.
for the post-translational modifications, we were talking about basically we're assembling the proteins, and what else?
You don't need to know details, but يعني in case you need to adjust the proteins. For example, if you need to add glycosylate the proteins or anything, so you need to add stuff to the protein. So this is post-translational modification. So after you translate the mRNA to protein, you modify the protein itself. We don't need to know details. خلاص. بس إنه يعني modifying the protein after translation.
Okay, thank you.
You're welcome. تمام. So we can move on to the test. It's just 2 questions. They're very simple, عادي. طيب, what is the correct order of the virus cycle events? In the chat, please. طيب, have 2 Ds, have a C, D. More answers? C. So it's between C and D. طيب, let's take them one by one. Have attachments in both, and then penetration and uncoating. So what comes first? After the virus attaches, will it start uncoating and releasing its genome, or should it penetrate the host cell and then do everything? First difference. Yes.
You attach and then you penetrate and then you have to
Exactly.
uncoat by the
Exactly. So you
degraded
penetrate.
enzymes.
Yes, penetrate and then uncoat, and then you have replication, assembly.
replicate.
Here, assembly and replication. Which one will come first? Will you assemble the virus before replicating, before making enough mRNA? You can't assemble it without having mRNA to put, without having RNA or DNA, sorry, without having genomic material, RNA or DNA to put inside the virus. So you need to replicate first and then assemble the virus. After you replicate everything, the proteins, the genome, and everything, and then you assemble the virus and then you release it. So first you attach, penetrate, uncoat, then replicate, assemble, and release. Got it? Yes, exactly. Correct answer is C. And here is the stages: attachment, penetration, uncoating, replication, assembly, maturation, and release. طيب, what is the function of early non-structural proteins? This is a very easy question. It was in one of the previous batch exams, so. A or C. تمام. Have A's, C's. طيب, early non-structural proteins will be used for genomic replication, basically. We said the immediate early proteins are for the initiation, the early proteins are for genomic replication, and the late are for capsid formation. But yes, most of you answered correct. تمام. Early non-structural proteins for genomic replication. طيب, now for the second lecture. Any questions before moving on? تمام. Now the enzymes. طيب, you need to understand those very well. You can have enzymes from the host cell or enzymes from the virus. تمام? Remember what we said about forming RNA? We said we have enzymes that should come from the virus itself. We said that there are enzymes that can be supplied by the host cell. Let's start by enzymes from the host cell. So here you have DNA-dependent DNA polymerase, DNA-dependent RNA polymerase. Now, to understand what they do. طيب, DNA polymerase will make DNA. So we're making DNA from what? DNA dependent. So DNA polymerase, we're making DNA from DNA. So this is normal. in human cells. عادي. We can make DNA from DNA.
Someone is asking you a question.
Uh-huh.
Someone is asking you a question.
Do we have genomic replication for request? Now we're going to talk about every group and what they do. Just listen to this part, and then if it's not clear by the end of this part, ask me again. We're going to talk about every group in detail. طيب, from the host cell. So we can make DNA from DNA. We can make RNA from DNA. So basically, anything DNA dependent from host cell, عادي. We have it. So DNA dependent DNA polymerase. Basically, we have one DNA strand. We're forming the second DNA strand. DNA dependent DNA polymerase. DNA dependent RNA polymerase. We have a DNA strand, which is the coding strand. We're making RNA from it by transcription. And then this mRNA will be translated into a protein. So those are the enzymes that are supplied by the host cell. Now we have enzymes supplied by the virus. The enzymes should come with the virus itself or made by the virus, but not from the host cell. Basically, here we have RNA polymerase that is dependent on RNA. So we're making RNA from RNA. This is the replicase enzyme. So here you have a positive strand of the RNA. You are making the negative strand in order to make more positive strands. طيب? This is the replicase enzyme. So this positive strand can be used normally to make a protein, but now you also need to make more protein, like of the genomic material in order to incorporate into the virus. Positive strand can be normally used to make the proteins, but now you need the RNA-dependent RNA polymerase to make more RNA strands. تمام? This is the replicase enzyme. Now you have, again, RNA-dependent RNA polymerase and negative stranded ssRNA. So you have a negative strand, you need to make the positive strand in order to make the proteins and to make more of the genome to incorporate into the devices. So this is, again, RNA-dependent RNA polymerase. We're making RNA from RNA, تمام? So this is for single-stranded RNA viruses. Positive strand, you need to make the negative strand in order to make more positive strands. Good. So those positive strands will be incorporated into the virus because this is a positive strand virus. Here you're taking all those negative strands in order to incorporate into the virus, and you're using this positive strand to make the proteins. Now, whoever asked that question about ssRNA, is it clear now? تمام. طيب. Now we also have something called reverse transcriptase, RNA-dependent DNA polymerase. Now, DNA polymerase, we're making DNA from RNA. تمام? Do we normally have this in the cell? In humans, are we making DNA from RNA? Okay, so here in this case, we have reverse transcriptase. Remember, we said we have something called retroviruses. So a retrovirus originally has RNA, but in its replication, okay, it's forming more RNA to make more viruses, but it also makes DNA. So this is reverse transcriptase. So transcription normally goes from DNA to RNA. In this case, it's going from RNA to DNA. So this is reverse transcriptase. We're going from RNA positive strand to DNA. Now DNA will form those double-stranded, and this DNA is going to be incorporated into the cell, and this will destroy the cell. خلاص, it's incorporated forever. تمام? But this is found in retroviruses. We're going to talk about them in a bit. But for now, just remember this. Those three enzymes are from viruses, and those two enzymes are from the host cell. Now, again, to the Baltimore classification. Baltimore classification, we said, first one is double-stranded DNA. And then those are found in adenoviruses and herpes virus. Herpes virus, we said, is the major group or the big family in the DNA viruses. And we also have adenovirus. Those two are double-stranded DNA viruses. Now, group 2 is single-stranded DNA. Now, this is a positive strand. A single-stranded DNA, and the example we said is parvovirus. We said DNA is usually double-stranded. The exception is parvovirus, which is single-stranded. This is group 2. Group 3 is double-stranded RNA. Remember, we said all RNA is single-stranded. One exception is reoviruses, which is double-stranded RNA. Good? Like, group 4, ssRNA, single-stranded RNA, positive strand, coronavirus. This is the major family in the RNA viruses. Now, we also have group 5, ssRNA, negative strand, orthomyxovirus such as influenza virus. So influenza A, influenza B, influenza C, those are orthomyxovirus. And remember that they are single-stranded RNA, negative strand, not positive. تمام? We have single-stranded RNA, positive strand with DNA cycle. retroviruses. So what enzyme do they have? They have the reverse transcriptase enzyme, which is RNA-dependent DNA polymerase. Those are the retroviruses. And finally, we have double-stranded DNA with RNA intermediate, which are the hepatinoviruses, such as hepatitis B. Now, those are the major classifications. You need to know the example of which viruses are in the group. تمام? Now, we're going to talk about every group and their replication. تمام? Group 1, double-stranded DNA, we said we have adenoviruses and herpes virus. Herpes virus is a big family. It has several groups under it. So under herpes virus, we have herpes simplex 1, we have EBV, Epstein-Barr virus, we have CMV, cytomegalovirus, and we have VSV, varicella zoster virus. Those 4 are under herpes virus. So if you get any of those are double-stranded DNA. If they ask you what is CMV genome, you say it's double-stranded DNA because it's a herpes virus. تمام? They're all under herpes virus. In double-stranded DNA viruses, this is easy, simple, straight to the point, because it's like in humans. So you have the double-stranded DNA. You take the coding strand, you make the mRNA, and then you make the proteins, عادي. And then the virus will assemble again and leave the cell. تمام? Double-stranded DNA.
But for the group 1 double-stranded DNA, you're using the negative strand to make the positive strand so that we can transcribe the protein, right?
Yes. Yeah. In this case, خلاص, you don't need to think about which one is the negative, which one is the positive. This is a double-stranded DNA. You're using it to make the mRNA. خلاص, يعني you assemble the virus. And this mRNA can be used to make proteins عادي. تمام? This is found in herpes viruses such as HSV, EBV, CMV, VSV.
So
Good?
I'm just a little bit confused. So here we transcribe the negative strand to make mRNA. And mRNA are positive strand. So why didn't we just use the positive strand that is originally okay, because we're
this is
changing
DNA. This is not virus.
this
is not
I just
RNA.
realized.
Yes, exactly.
Sorry.
Exactly. So you use the mRNA to make the proteins. You do not use the DNA. So you need to transcribe the DNA into RNA to make the protein.
So it's basically we're using DNA dependent RNA polymerase, right? The enzyme.
Exactly, yes. DNA dependent RNA polymerase, yes. تمام?
Yes, I got it. Thank you.
Now for double-stranded RNA viruses, we said the only RNA that's double-stranded is reovirus. And the reovirus we have rotavirus. Rotavirus is basically a virus found usually in daycare, so it will cause diarrhea in kids. So basically, if you have a kid with diarrhea, it's usually gastroenteritis caused by rotavirus. Now, in double-stranded RNA, you have the positive strand, negative strand of the RNA. We said we need to make proteins. تمام? So you use the positive or negative strand to make the protein. You have a positive strand now. فخلاص, you use this positive strand, you make the proteins. Now you need to make more of the negative strand too. So you need RNA-dependent RNA polymerase or the replicase enzyme to make the negative strand to make more positive strands. So now you can make the virus with the double-stranded RNA to incorporate into the different viruses. So you have a double-stranded RNA. You take the positive strand, you make the proteins عادي. The positive strand will also be used to make the negative strand. And now you can make more positive strands. You can make the double-stranded RNA. You can then surround them by the capsid, form the virus, assemble the virus, and then the virus is released. This is double-stranded RNA. So here you need RNA-dependent RNA polymerase, the replicase enzyme. Yes, you have a question? Okay. Someone is asking why make a negative strand when we already have one. Like we have one. Now, okay, you have one negative strand, you have one positive strand. You need to make more viruses. In the new virus, are you going to have one strand only? In the new virus, you're going to have the same thing as the original virus. The original virus had a positive and a negative strand. So you need to make positive and negative strands for the new virus. So you need to transcribe, you need to make the new strand too, to incorporate it to the new virus. So, okay, this negative strand won't help the original virus, but you need to make more viruses. Those new viruses will have negative strands too. Okay. تمام. This is for group 3. Group 4 is positive single-stranded RNA. Now, in this group, we have picornaviruses. again, remember the kind of viruses or the family of viruses in each group. So here you have coronaviruses. If they ask you what are those, they're single-stranded RNA, but they're positive strand, not negative. We said single-stranded RNA negative strand is orthomyxovirus, which is the influenza. We'll talk about it next. But for now, the coronaviruses, again, they have different viruses under them. You have poliovirus, you have hepatitis A, and you have rhinovirus. Have a question? What was group 2? Okay, we didn't talk about group 2 here because the doctor didn't touch upon it, but single-stranded DNA positive strand, which is a parvovirus. This is group 2. But for now, let's focus on the thing the doctor focused on. So this is the double-stranded DNA, double-stranded RNA. Now, single-stranded RNA, starting with the positive strand. We said for coronaviruses, we have polio, hepatitis A, and rhinovirus. If they ask you about any of those, so if I ask you what is rhinovirus, rhinovirus is under picornaviruses. So this is single-stranded RNA positive strand. Now, we have the positive strand. Remember what we said? Positive strand can be used to make the proteins عادي, quick. بعدين, you need to make the negative strand, and then this will form a double-stranded RNA intermediate. So this can be recognized by the cell. So the cell PRRs, which are basically the receptors that recognize that there's a pathogen or there's something wrong in the cell, there's something invading the cell. So this can be recognized by the cell. Now, in a single-stranded RNA positive strand, you have this positive strand, it will form the negative strand. This is the intermediate. This intermediate can be recognized by the host cell or the host PRRs or the receptors recognizing that there's a pathogen, there's something invading. Now, we'll go on with the replication, as we mentioned. RNA-dependent RNA polymerase used to make more RNA genome, which are used to incorporate into the new virus. This is group 4, the coronavirus, poliovirus, hepatitis A, and rhinovirus. All good? Group 5, again, single-stranded RNA, but negative strand. So here we have orthomyxoviruses, which are the influenza A, B, and C. What did we say? We said something special about influenza. What was it? Anyone remembers?
It replicates in the nucleus, not in the cytoplasm.
Yes. Yes, exactly. So we said all RNA viruses are usually in the cytoplasm and influenza in the nucleus. Tamam. Here we have the negative strand of the ssRNA. It's only a negative strand. Negative strand cannot be used to make proteins. So now we need to make a positive strand. Positive strand will make the proteins, and now we're making more negative strands in order to make the final virion. Tamam? Again, RNA-dependent RNA polymerase. Next will be group 6, the retroviruses. Now, under retroviruses, we have HIV. Remember this, it's important. Retroviruses is a positive single-stranded RNA, but it has a DNA intermediate. So it's a retrovirus. It can go from RNA to DNA. So what is the enzyme here? We have RNA-dependent DNA polymerase. We're making DNA from RNA, RNA-dependent DNA polymerase, which is reverse transcriptase, opposite of transcription. We're going from RNA to DNA. So here, the positive strand is used to make a DNA. This DNA is a provirus, so it can integrate into the host cell. Or this positive strand will make the mRNA or the RNA normally. When it makes the RNA normally, خلاص, it's replicating. This is replicase enzyme. Or it can form DNA. This DNA will integrate into the host cell genome, and it's going to be part of the host cell genome forever. Tamam? This is important in latency. We have a specific feature of some viruses which can undergo latency. So it will, it can either cause a chronic infection or a latent infection. We're going to talk about those patterns in lecture 3 and after this lecture. But remember that if it integrates into the genome or if it stays in the cell, it will cause infection for a very, very long time. طيب. We have a question. Do you mind recapping? طيب. Quick recap of all the groups. Double-stranded DNA. We're using the DNA to make mRNA. Normal. Nothing special about this. طيب. In this group, double-stranded DNA, we have herpes virus. No, this slide only? Oh, okay. طيب. Let's go to
I would
the last.
appreciate it if you recap it all.
all. طيب
Thank you
تمام
so
نرجع.
much.
طيب. هاي double-stranded DNA. ماشي? Another question? Me too. تمام. طيب, double-stranded DNA. We have the double strands of the DNA, positive strand, negative strand. Now, this DNA is used to make RNA. normal, nothing special. We don't need specific enzymes like reverse transcriptase, RNA-dependent RNA polymerase. Here we're just using DNA-dependent. So we can have this from the host cell. Under this, we have herpes virus. Herpes virus includes many viruses. We have HSV, have EBV, CMV, VSV, which are herpes, EBV, Epstein-Barr virus, CMV, cytomegalovirus, and varicella virus. Tamam? Group 3, double-stranded RNA. So here we have the RNA strands. We have positive and negative strand. Positive strand can be used to make the proteins, عادي. And the important protein we need here is RNA-dependent RNA polymerase. So this should be supplied by the virus because the host cell does not usually make RNA from RNA. So this should be supplied by the virus. And this is called the replicase enzyme. We're using the positive strand to make negative strand, making more positive strands. Group 4, positive ssRNA. RNA virus that is a single strand. This single strand is positive. This positive can be used to make the proteins. This positive strand can be used to make the proteins, and we can make a negative strand. This can be recognized by the host virus, the host cell, PRRs. And the positive strand will again be used to replicate, to make more of the genome, RNA-dependent RNA polymerase, which is the replicase enzyme. Under this, we have the coronaviruses, which are polio, hepatitis A, and rhinovirus.
But didn't we say we use negative strand? You said that positive strand we use it to make replication. Don't we use negative strand?
Replication can happen with anything. So here we have positive strand, here we have negative strand. You can use the negative strand to make the positive. You can use the positive to make the negative. But the difference is, in positive strand can be directly translated into proteins. Negative strand, you cannot use it to make proteins.
Yeah, but no, you said that we replicate, but no, don't we use the negative strand to make positive strand, more positive strand? I think you said here, you make.
It depends on the type of virus. فإذا الفيروس أصلا it has a positive ssRNA, to make new viruses, the new viruses should have positive ssRNA. All the viruses will be the same. But if the original virus is a negative strand, all the new viruses from that virus will have a negative strand.
Yeah. So we don't need to make a positive strand to replicate it. We can use the original strand. I think that's what I'm trying to say, because I got a little bit confused.
No, you're using the negative strand to make positive strand, then positive will make the negative. You can't use negative to make negative directly. So
I think it's either I misunderstood you or I think you did you did say something. No, we use the positive strand to make more positive. So that's why I'm just trying to, I think either I miscorrected you or you misspoke. It's either.
I'm sorry if I misspoke, but point is, if the original virus has a positive strand, all the other viruses will have positive strands. So you need to replicate. You need an RNA-dependent RNA polymerase to make those positive strands to incorporate into the new viruses. Tamam?
Yeah. But we need to make a DNA. We need to make a negative strand so that we can copy the positive strands.
Yes. But then in the actual exam, they usually don't ask about details about the replication. They usually care about the general group and what are the viruses in that group. They won't ask you about negative forming positive, forming negative, and all those specific details. Just remember the main enzymes involved. So RNA-dependent RNA polymerase, or DNA-dependent RNA polymerase, or reverse transcriptase, those enzymes. Those are the major things they usually ask about. They don't go into specific details usually. Don't worry about this.
Yeah, thank you.
Of course. Now, finally, for the last group, again, quick recap, HIV virus is a retrovirus. So you're using the positive RNA strand to make more of the RNA strands, or it can be used to make a DNA intermediate, which is by reverse transcriptase enzyme. This will form a provirus. This provirus can integrate into the host genome. This is all the major groups and the replication. So personally, that was the part I struggled most with when I was doing the viruses back in second year. So inshallah it's clear. Now for the next section. In RNA replication, you can have more base pair modification, meaning it's more prone to mutations, more prone to changes in the RNA. DNA is very neat. So when you replicate the DNA, you usually get the exact DNA you need. Meanwhile, in RNA, that's not the case. So you can have different things happening. You can have incorrect base insertion. So some enzymes are prone to making mistakes and they do not proofread. So you have RNA-dependent RNA polymerase and reverse transcriptase. When they form the new, like the new chain or the new genome, they usually insert some incorrect bases. So this will cause modification, and this will cause changes. So the final strand isn't always the same as the strand you exactly need. You also have two different cases happening. You can have RNA recombination and genetic reassortment. In recombination, you have two viruses infecting the cell. طيب, those viruses, when they both infect the same cell and they start replicating, sometimes you can have a genome having a part from one virus and a part from a different virus. So they combined again. So here you have a decombination. It will form viruses from the initial virus 1 and some from initial virus 2 and some recombinant viruses. So they have part of the genome is from 1 and the part from 2. So the same genome is going to have a part from 1 and a part from 2 in the same genome. Meanwhile, here you have genetic reassortment. Genetic reassortment happens in segmented RNA viruses. So here you have two strains of the virus. So you have strain 1 and strain 2 of this one virus. This is usually found in influenza viruses. Genetic reassortment can cause disasters. Now, what happens here is you have the two different strains of the virus infecting the same cell. This is a segmented RNA viruses. So now when we're forming the new virus, you will have different segments. So you have not one genome, one continuous genome, no, you have segments. So when you're forming the virus again, sometimes if two strains infect the cell, you can have mixing. So you have some from the red one, some from the blue one. It's not the same genome, but you have different segments from different strains. So this will cause a new progeny virus. This new virion could be a disaster because in case of influenza, for example, you have vaccines against this strain, you have vaccines against this strain, but now you have a new virus which has like powers from the original strain 1 and strain 2. This can cause a pandemic. This can cause disasters. So this will cause a pandemic because now you don't have vaccines, you don't have anything for the new virus with different segments. So in case of genetic reassortment happens in segmented viruses, it can cause pandemics. We're going to talk about this more when we talk about influenza.
So this happened in COVID, right?
Basically, yeah, it depends on the virus, yani, but usually pandemics happen with genetic reassortment in segmented viruses. I'm not sure about specific details, but yani, this is the main example you need to remember for genetic reassortment is influenza. تمام? طيب. How is it different from recombination? Yes, in recombination, as you can see here, the virus has one genome, and this genome is going to have, when two different viruses infect the cell, this one genome is going to have a part from 1 and a part from 2 attached together. So it's one genome with a part from each one of them. Meanwhile, in genetic reassortment, this is a segmented virus. So أصلا the virus has many different strands, many different parts of the RNA. Now, when you make the new one, it's going to take some from this strain, some from that strain, and you have a new influenza virus that has powers from both strains. Good? تمام. Okay, I don't think we have much more from this lecture. So recombination doesn't make a different strain. No, هلا. Wait. طيب, recombination, this will form a new virus, yes, but here you have like the same genome, but different parts in the same genome from different viruses. This will make, okay, you still have influenza, but it's a new strain. It has some from strain 1, some from strain 2. So this is a progeny virus. Now here you have examples, again, elaborating on the same point. So here you have antigenic drift and antigenic shift. So an antigenic drift is less dangerous than antigenic shift. Shift will cause pandemics. Antigenic drift will cause epidemics. So in a smaller scale. So in antigenic drift, basically you have a mutation. So the virus is replicating عادي, but there's a mutation in the virus. So here you have the same virus, which is basically, as you can see here, it had all the blue spikes. Here you have a mutation that caused a red spike. So this is minor. This will cause epidemics on a small scale. Meanwhile, in antigenic shift, you have dual infection. Now you have recombination. Recombination also can cause pandemics, so for whoever was asking. But this will form a new virus, and this is very bad. But you need to remember that recombination and reassortment differ by what's happening in the genome itself. So if they're replicating and mixing, now you have one genome with like parts from both. This is recombination. The assortment, on the other hand, you have segmented RNA or the segmented genome, and you have different segments joining into one virus. So this will cause pandemics, and this is very bad. But the major example you need to remember is in reassortment and influenza. Like, again, as I mentioned, influenza. So it can undergo antigenic drift عادي. It can have a mutation in one strand, and this will cause a small scale epidemic. But in pandemic, you have antigenic shift. Antigenic shift in which you will have like different segments. Yes, you have questions? Are they not what should put? Yes, as I mentioned, if it's a mutation only, mutation will cause a drift. But if you have a mixing of the genome, this will cause a shift. But in this case, here you have a shift because there's mixing of the genome in different strains and species, and then when they jump to humans, they will cause a pandemic because now you have a whole different genome happening. And they can also cause, there will be mixing basically between the different species. So you have avian, you have swine, you have human. So they will be mixing. As you can see, you have segments from different strains affecting different species. And then again, this can cause pandemics. So yeah. Now, speaking more about influenza. Influenza has RNA and it's segmented and it has the capsid, it has everything. Now we have something called, and now here you have the matrix proteins, everything, but the spike proteins, you have two. You have hemagglutinin, which is H, and you have neuraminidase, which is N. H will help the influenza bind to the target cell, to the host cell. N will help it escape after replication. So H for binding, N for leaving. Now you have different types. You have different different types of H. So you have numbers, different numbers. So you have H1, H2, H3 for different types of hemagglutinin. Again, you have N1, N2, N3 for neuraminidase again. So antigenic drift could be caused by some changes in like the H and the N. Now, major changes will cause different strains of influenza to appear. Now, why do we care about the different strains? Because the vaccine for influenza will be targeting those spike proteins. They will be targeting the H and the N. They'll be targeting the influenza depending on the type of H and N. So, for example, you can have influenza that's H1N1 or influenza that's H1N5. So we are worried about some specific strains that have potential to cause pandemics. So we need to see every season what are the strains of influenza that are appearing. Because if we are worried about that specific strain, it can cause a pandemic. So you need to monitor those strains because some of them you can give a vaccine. So, and you can formulate the vaccine according to the type of H and N on the influenza. So if it's H1N1, we are worried because this can cause a pandemic. If it's any other type, I don't remember the specific numbers, but some types do not have pandemic potential, but we are worried about some that can cause pandemics. This is an example of drift. Drift and shift are about what is happening in the genome. Now we're talking about the different spike proteins and how we're using them to make vaccines. When we're talking about drift, the major thing you need to know about drift is a mutation in the DNA. Shift is mixing. So here you have mixing either in one genome or you have mixing in different segments and so on. So when you want to differentiate between drift and shift, you need to look at what's happening with the genome itself. And when we're talking about pandemics, we're talking about shifts. Type latent infections. 1 second. Latent infections. Remember we said we have some viruses that can stay silent inside the cell. So if they stay silent, they are currently not causing infection. They are currently silent, but they can later on decide to start replicating and cause infection. How do they decide to start replicating? Basically, if there's any stressors, any cytokines, any inflammation that could trigger the virus to start replicating again. Now, when do we have latent infections? If the virus can integrate or stay in the cell as a plasmid, or it can be integrated into the genome itself. Remember we said when it's integrated in case of retroviruses, when it can form a DNA intermediate. So it's RNA, forms DNA. This DNA is now incorporated into the host cell genome itself. So here, when it's integrated, it can cause a latent infection, so it can stay inside the cell. And whenever this genome is replicated, to form more of the viruses. As plasmid, for example, you have herpes. Herpes simplex virus, it can stay latent in the trigeminal nerve ganglion. So this is the trigeminal nerve. It can stay as a plasmid in the ganglion of the trigeminal nerve. So it can either stay as a plasmid, so those circles you see as episomes, and it will stay latent, or it can incorporate into the host cell itself. So this is the case of HIV. And in some specific cases, it can get reactivated again. Again, as I mentioned, in case of infection, inflammation, and stressors, cytokines, anything triggering the virus to start replicating again. Finally, for this lecture, we have satellite viruses. Satellite viruses are very tiny, and they need help. They can't like just do their own thing alone. They need another virus to help them. So satellite viruses are tiny and dependent. They need a helper virus that's co-infecting. So those will infect the cell together. And now the tiny virus, the satellite virus, can take action. And we have different examples. We have 3 examples here. We have a parvovirus, which needs a helper virus that could either be adenovirus, herpes virus, or vaccinia virus, in order to help parvovirus take action and start replicating and do its own thing. And what happens in this case, in this infection, you have aplastic anemia and teratogenicity. Teratogenicity is basically when it infects the mother, and then the mother will pass it to the fetus through the placenta, and then the fetus will be affected. So this can affect the growth of the fetus, cause mental retardation, intellectual disability, and so on, depending on the type of virus. So parvovirus is a satellite virus, needs adeno, herpes, or vaccinia virus, and it will end up with aplastic anemia and teratogenic. Virus. Okay, so yes, in this case, adenovirus and herpes virus, as you correctly mentioned, are a double-stranded DNA virus, but they need those specific viruses. And you don't just say, okay, they need DNA viruses. They need those viruses. So, adeno-associated virus, again, will need the same helper viruses. You have adenovirus, herpes virus, vaccinia virus, and they will cause, the genome will integrate into the virus, and it's used as a vector for gene therapy. You don't need to know details. I don't remember when they asked about this last one, but just memorize this. And they don't usually ask in detail about this. Finally, you have hepatitis D virus. Hepatitis D needs hepatitis B infection. So hepatitis D by itself won't infect the cell, but if you have a hepatitis B virus, hepatitis D can use its antigens and infect the cell. So now prognosis will be worse when the liver is infected by both B and D. So hepatitis B by itself, if it affects the cell, it can cause hepatocellular carcinoma, can cause many things, but it usually just causes an acute infection and disappears. But if it has a hepatitis D infection with it, the prognosis is worse. That's all you need to know for this second lecture. So we talked about RNA and different mutations that can happen, different combination, reassortment, influenza virus, and how we use the H and N serotypes to make the vaccines. We talked about latent infections, satellite viruses, and all the different groups and their genome.
Can you please repeat the N and H slide? Just briefly. Thank you.
Yes. طيب. Here you have spike proteins on influenza virus. Influenza has H, which is hemagglutinin, and N, neuraminidase. H will help it bind to the cell surface and will help it escape after it finished replication and everything. Now, those H and N, because of antigenic drift, they will keep changing. So you have different serotypes of H, different serotypes of N. So every season you have different kinds. So the influenza could either be H1N1, H2N1, H3N2, and so on. So you need to track the differences. You need to track the H and N in order to know what vaccine to use, because the vaccine depends on this the specific serotype of H and N. تمام? So you need to track those in order to make the vaccines. You have some specific strains which can cause like pandemics, and they can cause like serious stuff. So you need to make sure the influenza is not of those serotypes too. So usually you need to know what are the serotypes, what type of influenza you have this season in order to make the correct vaccine for that. Because you can't use the same vaccine for everything. You have different receptors, you have different spike proteins on different influenza. Got it?
Yes, yeah, thank you.
Can I ask you a question? So an example of a shift is basically when we're having an influenza virus that's being transmitted from the animals to humans, basically, and as they are being transformed from one animal to another animal, a new genome is being added to it, and that's leading to pandemic?
So what you need to know here, as I mentioned, to know that this is shift or drift, or what's happening is the mixing of the genome. So shift means the mixing. So you have a strain that usually infects pigs. You have a strain that usually infects humans. Now, when they co-infect one cell, you have a progeny influenza virus that has genome from the pig strain, from the swine strain, and genome from the human strain. Now they mix, they make one new influenza virus with different segments. Now this is an antigenic shift. This will cause pandemics. So the main thing here, main thing you need to remember is that you have mixing of different strains co-infecting one cell. Come on.
Thank you.
You're welcome. Now, a few questions. What enzyme is found in retroviruses needed for the formation of double-stranded DNA intermediates? In the chat. Yes, exactly. So it's C, RNA-dependent DNA polymerase. We're making DNA from RNA, and the other name, as someone correctly mentioned, is reverse transcriptase. Yes, exactly. Next question. Influenza B is okay. If you guys want to send in private, so others won't know the answer. بس عادي يعني. Okay. Yes, exactly. So influenza B is a single-stranded RNA that is a negative strand. Correct. Now, lecture 3. Disease. طيب. We have two terms. We have pathogenicity and virulence. Pathogenicity is basically those okay, yeah. طيب. The first sentences here are what's included in your lecture, in your slides. So pathogenicity is, as the doctor mentioned, yes, one question. Can we have a break? Does everyone want a break? Do you guys want a break or keep going?
Is the second part much longer?
We finished half. There are 4 lectures, we finished 2. And we have 2 left. So, no break. خلاص majority ask for no break. 5 minutes. Host, can we have a 5-minute break? Just quick break and we'll be back. تمام? Okay, now we can continue with the second half. Like, starting with defining some terms. We have pathogenicity and virulence. Now, pathogenicity is basically the severity of disease caused by different microorganisms. So here, pathogenicity is basically the ability to produce disease. So a pathogenic organism will be able to cause disease in the host, like human, for example, or the host cell. Meanwhile, non-pathogenic will not cause disease. So keep in mind that usually most infections are subclinical, so they will be asymptomatic, they're not causing disease, but disease could be caused either by cytolytic or immune-mediated effects. So either the virus can itself have an effect on the cells, so itself can destroy the cell, it can affect the replication of the cell, it can cause something, or immune-mediated. So the immune response to this virus will cause the disease. So for example, when the immune response is activated against the virus, it will cause fever, it will cause symptoms, and so on. So the disease could either be by the cytolytic effect of the virus itself or immune-mediated. But keep in mind that most diseases are usually, or most infections are usually subclinical, asymptomatic, and they do not really cause infection. Now, for virulence is the severity of disease caused by different strains of the same microorganism, measure of the infectious dose. Basically, in simple words, it's the disease-producing power or the degree of pathogenicity, measuring the degree of pathogenicity. So in virulence, you have different strains, and they can cause, and different strains can cause infection, but you need to know the disease-producing power, the degree of the pathogenicity. This is achieved by different factors. So, for example, those factors are called virulence factors. So it could either be a capsular antigen, it could either be exotoxins. For example, exotoxins produced by some. In this case, for example, you have bacteria, but virulence factors can also be for viruses. So those factors are basically specific aspects or specific descriptions of the infecting organism or the infecting cell that will allow it to cause the disease. So virulence is the or the degree of pathogenicity, the power of the cell to produce disease. Pathogenicity is basically the ability to produce disease. تمام؟ طيب. Now we have two other terms to define. We have productive infection and we have an abortive infection. Now, a productive infection will cause disease. So this will depend on the infectious dose and the state of the cell. So a productive infection, so basically you have viruses affecting the cell, they're infecting the cell, infecting the host, and they will cause a disease. So it is a productive infection. You have enough like viable viruses, enough viruses in order to cause the disease. Meanwhile, you also have something called abortive infection. In abortive infections, you do not have enough infectious virions. So, as you can see here, this is a microscopic image of hepatitis B virus. So those spheres, as you can see, they have like the surrounding capsid, and they also have the genomic material. So this is viable. This can cause infection. This is an infectious virion. Meanwhile, you also have those filaments. Those are non-infectious surface antigen particles. So they only have the surface antigen particles, but they do not have the genome. They cannot cause disease. So if you have too many of those and not enough of the viable viruses, this will cause an abortive infection. It will not cause a disease. What is the significance of non-infectious surface antigen particles or those without the genome? They can be used to make vaccines. So those vaccines, they are attenuated viruses by continuous passage. So basically, you keep culturing those viruses in the cell culture, and then you will take those surface antigen particles or those like basically shells, and you'll use them to make the vaccines. Those will only have the surface antigens. As we said, the antigens can be used to make the vaccine without causing the disease itself, because those cannot replicate and cause the disease. You're only using them in order for the body to produce antibodies against this virus, and this will protect you. This will protect your cells without causing the disease itself. And here again, you have another picture. Here you have an empty capsid. It does not have a genomic material. This is an infectious virion. So here you have genomic material with the capsid. So it's basically the amount of infectious virions is the thing that will determine if the cell or if this infection will cause a disease or will not cause a disease. Now, speaking of diseases, you have different patterns. Okay, we have a question. طيب, what does infectious dose mean? Okay, so it's basically the amount of viruses that is needed in order to cause an infection. So if the virus has a high infectious dose, it means you need so many of those viruses in order to end up with a productive infection causing a disease. But if a low infectious dose, only a few viruses are enough to cause a disease. Good? Sorry for not specifying or explaining earlier. I hope it's clear. Okay, good. Now for the disease patterns, you have 4 different disease patterns in viral infections. You have acute, you have latent, you have chronic, and slow infections. Let's start with acute. Acute, straight to the point. The virus will replicate, so it causes the disease. Now, you will have immunity against the virus, you're done with the infection. That's it. So here you have signs of illness due to cytokines. So we said the body will start fighting the infection, fighting the virus, the viral infection. It will release cytokines, it will release immune mediators, and this will cause the signs of the illness. So basically, the fever is caused by IL-1, TNF alpha, you have redness, you have coughing and everything. So all those are caused by the immune response. You also have memory B cells formed. So after this infection is done, so when you have the host attacking the virus, attacking this infection, it will form memory B cells in case you get infected by the virus or by the same pathogen. Later on, those B cells will start replicating fast. It won't allow this virus to cause a serious infection. They will attack this virus again. You're done with the infection. So in acute, here you can see the virus is replicating, and then you're done with the infection when there's immunity. Where do you see this disease pattern? In influenza virus and hepatitis A. So usually, hepatitis A and influenza virus will cause acute infections that will get resolved. Clear? Clear. Now you have something called latent infection. So first, you have an acute infection, عادي. You have virus replicating, causing an infection. Now it will start, you start clearing the infection, but here you have latent infections which can be silent. You see this dotted line? Here you think you have nothing, you have no symptoms, you have nothing going on, but you actually still have the pathogen, you still have the virus. So even in this latent stage, you can still have infective virions even if you're asymptomatic. When does this happen? So in latent infections, you have this in the herpes simplex viruses, basically the herpes family. It can include herpes simplex. It can include CMV, EBV, and varicella virus. So those usually cause latent infection. So it will be latent inside the cell. Remember, we said HSV can be latent in the trigeminal ganglion. It can form the episomes, the circles, and they will stay latent. And then it will be reactivated by UV or stress or hormones or cytokines. So it's either affected by the environment or by the host cell itself. And it will get reactivated. And now it will cause infection again later on. So for example, in varicella, usually this infection will be in kids. It will cause chickenpox. Varicella will cause chickenpox. It will cause chickenpox. خلاص, you're done with the infection. It subsides, but you still have varicella inside your body. It will be latent. You do not have any of those vesicles, any of those bumps of the chickenpox infection, but you still have the virus itself. It can get reactivated later when you're 60 as shingles. So in shingles, you will have reactivation of the varicella virus that was latent, but now it's activated due to hormones, cytokines, stress, whatever, and it will cause shingles. Okay, so here we said we have the herpes family. You have also HIV, which is a retrovirus. It will be activated again by NF-kappa B, which is basically responsible for inflammation. So in case of inflammation, HIV will be deactivated. Herpes family, again, as I mentioned. Yes, we have a question. Can't infect other people during latent speed. Yes, we said a lot of infective virions, even if you're asymptomatic. So you can still transmit the infection. تمام? This is for the second pattern, the latent infection. You have a chronic infection. What is the difference between a chronic and a latent infection? Does anyone know?
Chronic infection is basically going to stay for a very long time and it's going to persist, basically.
Yes. So in latent, when in the latent phase, you won't have any signs of the infection. You do not have any symptoms. In chronic infection, the infection is not fully cleared. So it stays in the DNA either as in the host DNA, it stays integrated in the DNA, or as episomes, and it will stay and keep on going. It's a chronic infection. and the carriers will be spreading the virus whether they have symptoms or they do not have symptoms. So the chronic infection is not cleared. It will stay in the system or in the host, and this is found in hepatitis B and HIV. So HIV will keep on going. It will keep on infecting the T cells, and at some point it will cause AIDS, acquired immune deficiency syndrome. So hepatitis B can cause an active infection and then a chronic. It will keep on going. HIV again, it will keep on going, and at some point it can also cause AIDS. So it infects like a huge amount of the CD4 cells. So those
Can I ask you a question?
Yes.
In COVID-19, which is like what happened in 4 years ago, some people were asymmetric and they they don't have any symptoms, and some people after the disease they continued having these symptoms. So is COVID-19 considered part of the latent infections or chronic infections?
No,
It's just
this is not. Don't confuse yourself with like such details. The difference in symptoms is basically because different people have different immunity. So if you have good immunity, you fight the infection. Other people cannot fight the infection, and they would stay symptomatic and they show symptoms, and later on, like many patients died because of COVID. But for now, we're just talking about the kind of infection that will stay for a very long time. For example, latent infection, such as herpes. So herpes will stay. For example, a kid will be infected, and then later on due to another infection, inflammation, and so on, they will start getting cold sores. Those cold sores are basically a sign that you have reactivation of the herpes virus. Again, with varicella. Varicella basically will cause chickenpox in kids. It will be latent, خلاص, you don't have any symptoms. Again, it will be reactivated later on, and it will cause shingles. How is HIV chronic and latent? So, okay. HIV can be integrated in the genome and it will cause a chronic infection, or it can be latent. It does not show any symptoms. Later on, it gets reactivated and will cause a chronic infection. But the major thing you need to remember for HIV, if you get asked this question, just remember that HIV will cause a chronic infection. This HIV was mentioned in your slides in the transcription in both latent and chronic infection. It was mentioned in both slides, but in the diagram here, it's mentioned in the chronic infection. So the major thing you need to remember, latent infection for the herpes and the chronic infection for hepatitis B and HIV. But according to Dr. Garwin, it was mentioned in both. طيب. Finally, we have can this come in OSPI? You rarely get such questions no, actually it can come. I'm not sure, honestly. I don't remember if we had it in our OSPI, but you just study it just in case, and you studying it either ways for the MCQs. And yeah, just remember what kind of viruses will cause latent, acute, chronic infections, and so on. Because they usually get very creative with the OSPI questions. I think you guys have another PAL session for OSPI, صح? I'm not sure who's giving that session, but inshallah they'll be covering everything. All the high yield stuff. طيب, the final disease pattern is a slow infection. So this slow infection, as you can see, this is a dotted line. So it will incubate for very long years with no symptoms, but it's still infectious. You're still passing the prions. In this case, it's a prion disease, which is basically a weird protein. So this protein will keep on like incubating for years. You won't have any symptoms, but you're still infectious. You can still give pass infection to other people around you. It is progressive viral disease of the CNS. So for years you won't have any symptoms, and then suddenly they will show some symptoms. And this is a very slow infection, so it does not cause acute infection at first, and then it will be latent or cause a chronic infection. It will be very slowly, very progressive, and then suddenly it will show infection later on, after years. And this is the prion disease. Prion disease has other names. You have Creutzfeldt-Jakob disease. You have other names. It's written in the notes section. I'm gonna send you the slides, and you can read everything. تمام? So those are the disease patterns. Now we can move on to oncogenic viruses. Oncogenic viruses was also explained in another class session about neoplasia. it was in more detail about the specific viruses and the way they infect the cells. But here we just need to know which viruses infect which cells to cause which cancer. تمام? I'm only gonna cover the things Dr. Garn like mentioned in the slides. So first thing we have, you need to know that oncogenic viruses will cause cancer. First one, let's talk about P53 and retinoblastoma. P53 and RB are part of the cell cycle. So they are basically a checkpoint that will make sure that everything is going well for replication. You have several viruses that when they affect the cell, they will affect P53 and retinoblastoma, and this will affect the cell cycle replication, and it will cause like excessive replication, because usually the dose will stop the cell from replicating. There is something wrong with the DNA. But now you have viruses infecting, they will affect those checkpoints in the cell cycle, and they will cause a disease. And in this case, they will cause cancer, because very rapid replication due to unregulated cell cycle will cause cancer. And what viruses can cause this due to affecting P53 and retinoblastoma? You have SV40, which is a polyoma virus, and it affects the humans and monkeys. You also have adenovirus, and you have papillomavirus, which is HPV. HPV has different proteins. You have E6 and E7. E6 will affect P53. E7 will affect retinoblastoma. Again, you have more details in neoplasia lecture in pathology. For now, just remember that you have HPV, which is a papillomavirus protein, papillomavirus. You have adenovirus and SV40. Those will affect P53 and retinoblastoma to cause cancer. Now, second one is targeting B cells. Now, which virus will target B cells? You have EBV, Epstein-Barr virus, also called as HHV4, human herpes virus 4. This will affect B cells, cause B cells to divide and replicate uncontrollably, and this would result in different cancers. EBV can cause Burkitt's lymphoma or nasopharyngeal carcinoma. تمام? So this is the second oncogenic virus, EBV. It can affect B cells, causing Burkitt's lymphoma or nasopharyngeal carcinoma. Third one we have is HHV8, human herpes virus 8. This will affect endothelial cells, mainly in HIV patients. So usually HIV patients, if they're affected with HHV8, it can result in Kaposi sarcoma. Kaposi sarcoma is usually a skin lesion, as you can see here. HHV8 will affect endothelial cells, resulting in Kaposi sarcoma. You don't need to know much details for it. Yes.
But what does HHV8 stands for? بس I forgot.
Human herpes virus 8.
Okay. And human herpes virus 4 is going to lead to Burkitt's lymphoma?
EBV. Yes.
And
Burkitt's lymphoma and nasopharyngeal carcinoma.
both of them, right?
Yes, it depends. It depends on the case.
Okay.
But usually this virus can result in those two carcinomas or lymphoma.
Thank you so much.
You're welcome. But now we have more. You have viruses targeting T cells. HTLV1, HTLV2, which is human T lymphotropic virus 1 and 2, affecting T cells, and they will cause lymphomas. You also have hepatitis B, hepatitis C, or from the name hepatitis, so it will affect the liver. It will cause liver cancer, or it can cause a chronic infection. Remember we mentioned hepatitis B and also hepatitis C. They're both involved in chronic infections. So they will cause a chronic infection, but now we're discussing oncogenic viruses causing cancer. So one of the results of hepatitis infection is liver cancer. So HTLV will affect T cells, causing lymphoma. Hepatitis B and C affect liver cells, causing liver cancer. HHV8 affect endothelial cells, usually in HIV patients, causing Kaposi sarcoma. Here you have EBV affecting B cells, causing Burkitt's lymphoma and nasopharyngeal carcinoma. And we said those viruses, SV40, adenovirus, and papillomavirus, can affect P53 and retinoblastoma in the cell cycle. Now, we're done with oncogenic viruses. Moving on to congenital infections. Congenital means they pass from the mom to the fetus. تمام? So this is called vertical transmission from the mom to the fetus through the from the mom to the fetus will cause congenital infection. It can either be during pregnancy through the placenta, it can be intrapartum, it can be postpartum, vertical transmission. So, during pregnancy, what can cross the placenta? What crosses the placenta, for example, rubella, CMV, and HSV. So those are known as torch infections. Torch infections are basically like infections that can affect the fetus and they're teratogenic. So they can be passed across the placenta in the first trimester. So if the mother is infected by those viruses in the first trimester, she can pass the infection to the fetus through the placenta, and this will cause specific problems in the fetus. For example, rubella will cause intellectual disability, it will cause cataracts, hearing loss, and so on. Cytomegalovirus will cause microcephaly, it will cause calcifications in the brain, HSV2. So those will cause infections in the fetus during pregnancy. Intrapartum means during birth. So it's through the birth canal. And this can be caused by hepatitis B infection. So while the baby is being born, if the mother has hepatitis B, she can transmit this infection to the baby. So what we do is we vaccinate the baby as soon as they're born. So you have we're going to talk about vaccines later on. So vaccines are scheduled at different times in the baby's life. Hepatitis B is given at different times, and first vaccine is given at birth. Postpartum, how can the mother pass infection postpartum to the baby? Through breastfeeding. And the main culprit here, main virus, is HIV. So HIV is passed through breast milk from the mother to the baby, postpartum. Those are the vertical transmission from the mother to the baby or to the fetus. Now we have other forms of transmission. Everything, again, is explained in the notes section, like in detail if you want. Now you have some viruses. For example, here you have influenza, coronavirus, and rhinovirus. They affect the respiratory system. So they will go through the nasal epithelium and cause the infection. Influenza, corona, and rhinovirus. You have other infections that are through direct transmission, either through skin lesions or through saliva. So through skin lesions or saliva, you have the herpes, which is HSV1. As we said, it can be transmitted through kissing, unlike HSV2, which is through intercourse. So here you have HSV1, you have CMV, EBV, varicella. Those can cause infection through skin lesions or saliva. You can have through blood. Through blood can be HIV, hepatitis B, and hepatitis C. Now, you need to remember hepatitis B because hepatitis B vaccine is also needed for healthcare workers. So hepatitis B can be transmitted through blood. So for example, through syringes, needles, and so on. So if you have a hepatitis B patient and you get pricked by a needle from that patient, you can get hepatitis B too. So you need to take the vaccine as a healthcare worker. Now you also have fecal-oral infections. So fecal-oral, you have rotavirus, have hepatitis A, and Coxsackie virus. So those can cause fecal-oral transmission. We mentioned rotavirus in daycares, remember? So we said like children who go to daycare, they usually have fecal-oral transmission of rotavirus, can cause gastroenteritis and diarrhea. Again, hepatitis A can cause traveler's diarrhea, and you have Coxsackie virus. You also have this question, virus. Yes, we're going to talk about this later on, but yes, exactly. Now, you also have sexual transmission, which is HIV, HSV2, herpes simplex virus 2, hepatitis B, and HPV, human papillomavirus. Those can be transmitted through sexual intercourse. Finally, you have rabies and arbovirus, which are zoonotic infections. So we said zoonotic can jump from species to species. Here we have animals. Rabies can be from rabid animals. You have dogs, dog bites, and so on. Arbovirus is through mosquitoes. So you have insects, arthropods, mosquitoes, such animals or such insects can cause arbovirus infection. Those are different forms of transmission, plus the congenital ones we mentioned before. Now for testing. Testing is, I believe, the last section for the third lecture. So we're almost done with the third lecture. For testing, you have different rules for virus testing. When do you do this? You do it when you suspect an epidemic or a communicable disease. So if you suspect that this virus could actually spread to many people, cause a disaster, cause an epidemic, or it can be very easily communicable between people. So you need to test it. This is number one. You need to get a sample from the correct site ASAP, and you get a blood sample too, and you grow this in tissue culture. Why do you grow this in tissue culture? We said viruses need a host cell. It can't grow on its own. So you can't just take a sample and just let it grow by itself. The viruses can't grow without a host cell. So you grow it in a tissue culture. In those pictures, you can see this is a culture for fibroblasts. Fibroblasts are elongated, spindle-shaped. Here you have epithelial cells. So those are different shapes of appearances of the culture, basically. Those are tissue cultures. Now you have different approaches. Okay. Here you have isolation. You have viral nucleic acid, antigen or virus itself, and you have serology. We're going to talk about them one by one. Let's start by isolation. So you're isolating the virus, putting it in the tissue culture, and you are observing the cytopathic effect of the virus. Cytopathic means characteristic features or changes that occur in the infected cell. So you have different appearances of what could happen in the affected cell. For example, you could have nuclear inclusion bodies. So those are nuclei, and you can see some weird spots inside the nuclei. So those are called inclusion bodies. If you see this appearance and they ask you what is the cytopathic effect, this is nuclear inclusion bodies. Here you have vacuolation. So this is the cell, and you can see kind of vacuoles forming in the cell. So this is vacuolation. Here you have syncytia, where basically you can imagine multiple cells joining together. So you have a multinucleated giant cell. You have a giant cell with a million nuclei. So this is a syncytia. And you also have cytoplasmic inclusions. You can see inclusions in the cytoplasm, and you can see a multinucleated giant cell. Here you have plaque formation. So here what's happening is the cells are dying, and those dying are forming plaques on the tissue culture. So those are necrotic dead cells. This is multinucleated giant cell, but you also have cytoplasmic inclusions in this picture. Here you have vacuolation, and here you have nuclear inclusion bodies. Now, to measure the infectious dose, basically to know the amount of viruses in the infection is. Can you repeat? Tamam. This is cytopathic effect. Cytopathic effect means what is the appearance of the cell culture due to this viral infection. So viral infection can result in different appearances of the culture. So one of them is like the plaque appearance. The plaque is basically the dead necrotic cells forming plaques in the culture. So this appearance is plaques, dead cells. Here you have syncytia, where basically you have a multinucleated giant cell, one big cell with many nuclei. You can see also nuclear inclusions in the nuclei, and you can see cytoplasmic inclusions in this picture. But this is a multinucleated giant cell. Here you have vacuolation. You have one cell with vacuoles in the cytoplasm. So this is vacuolation. And this is nuclear inclusion bodies. So in the nuclei, you can see inclusion bodies inside, those weird dark spots inside the nuclei. That's it. To measure the infectious dose, you do a plaque assay. So basically, you take the tube, and which has this. So it's just like empty sacs in the cytoplasm. A plaque assay, basically, here you have a tube with the cells, and you have the viruses too. Plaques, as you can see here, we said plaques are basically dead, like the dead cells. So this is the appearance of a plaque, and this is uninfected control. The cells are replicating normally. Nothing is affecting their replication. They're growing normally, and this is a plaque. What happens is here you do dilution, because if you take a culture of this by itself without diluting it, this is what you're going to see. You're going to see so many plaques in the plate, because you have a lot of viruses. So when you culture it on that plate, you're going to see a lot of plaques. You can't count them, so you can't quantify the amount of viruses in that sample. So what you do is serum dilution. So you keep diluting it, and every dilution, you'll get less and less viruses. So here they're too numerous, and then when you dilute it further, you can see 16 plaques. If you dilute it more, you can see 4 plaques, and now you can count them, and now you can know the amount of plaques, or the infectious dose of the virus, sorry. So here, for example, you can count 4 plaques at this dilution, 10 to the power of -7. if you have a high infectious dose, basically you have more plaques with even with the like more dilution, you're gonna see a lot of plaques. But sorry. So what you're doing is you're trying to achieve a dilution in which you can count the individual plaques. تمام? Clear? Sorry. طيب. Okay. Now you have microscopy. So we saw them on tissue culture by putting them in like cultures after dilution. We can see them under microscope now. Other than counting the plaques, you can also visualize them under the microscope. Microscope fluorescent, but basically you add dyes or you add unlabeled antibodies, which will like give a color under fluorescence, and you can see them, you can visualize them. You don't need to know them in details. Just remember, microscope could be electron microscope or fluorescent. So we did after approach one. Yes, so here you isolated them and you're looking at the appearance of the cells. Here you're counting the amount of plaques, so it's different. Here you're just observing what's happening with the cells. Here you're trying to quantify, you're trying to measure the amount of the infectious dose. تمام? So in low infectious dose, you can have a few plaques. In high infectious dose, you can have many. Here in microscopy, you're just trying to see the appearance of, so each one has a different purpose. Here you're trying to see the virus itself. Here you're trying to count to know the amount of the infectious dose. Here you're trying to see the appearance of the like the cells affected by the virus. Now we also have molecular techniques. That's what I mean to you should say. Microscopy. Yes, yes.
Are the pictures of the cytopathic effect important?
Yes. So remember them. Remember what
Thank you.
remember what they show. Here you have molecular techniques. You have different techniques. Again, those are more in biochemistry. You don't need to know them in detail, but we're just like touching upon them for the sake of completion. Here you have a reverse transcriptase assay or RT-PCR. Reverse transcriptase PCR, basically, you form the complementary DNA from the RNA, and then you do PCR, polymerase chain reaction, in order to increase the amount of DNA. And now you can use this to like do many tests. So for example, we know PCR for COVID, PCR for many viruses. Basically, you're trying to isolate the genome and you increase the amount in order to run some tests on it. Gel electrophoresis for amplified products. They can be, you can do electrophoresis for the RNA itself or for the proteins and see the way it's moving on the gel with the electric current. Or you can do sequencing to know the sequence of amino acids coding for that specific virus. So those are just different techniques to identify the virus. تمام. And finally, we have serology, which is quantitative ELISA. So in quantitative ELISA, here you have different, basically wells, and if this is a positive control and this is a negative control, here you have like the sample with the viruses, and this has nothing. Basically, you keep diluting it until you stop seeing anything. So we're starting from here. Now this is very dark. So this has a high amount. And now you keep diluting it until you stop seeing anything. And you will count the titer. You will see the titer to know the amount of the viral load in that sample. So do you think this sample will have a low amount or a high amount? Second one.
Low amount?
Yes, this has the lowest load. So because after only 2 dilutions, you stop seeing any color. Also, you have this has the lowest load of what? His serology is using ELISA to check the antigens of the virus or to check antibodies against that antigen. So basically, you're using them to know the amount of viruses you have. You're not visualizing the virus directly, but you're using antibodies and antigens in order to know the amount of viruses you have. So if you stop seeing after only 1 or 2 dilutions, this has the lowest dose. After many dilutions, this has the highest load. So you need multiple passages. You need multiple dilutions in order to stop seeing the color. تمام? This is quantitative ELISA. This uses serology. So this will utilize antibodies in order to check the antigens for the viruses. تمام؟ طيب, this brings us to the end of lecture 3. One more lecture to go. Now we have a few questions. Which of the following causes chronic infection? تمام. Yes, everyone's right. E. Hepatitis B causes chronic infection. Hepatitis A causes what?
Acute.
Hepatitis A causes acute infection. تمام. Next question. What is the target of HPV, enabling it to cause cancer? In other words, why is HPV oncogenic? What does it affect? Okay, we have a few answers. Yes. HPV has E6 and E7. E6 and E7 will affect the cell cycle regulation by affecting P53 and retinoblastoma. And finally, which molecular technique helps detect antigens in the titer to quantify the viral load, to know the amount of viruses you have using antigens, by detecting antigens? Yes, Eliza. Finally, last lecture. Any questions before we proceed? So, nothing? We can move on. Yeah, me too. So, classification of virus kingdom. Now we're going to talk about classification in different ways. Let's start by classifying into DNA and RNA. We said we have different viruses or different families under DNA and different families under RNA. The biggest or the major family in DNA viruses is herpes virus. Major family in RNA virus is picornavirus. This does not mean we do not have any other families. We still do, but those are the biggest ones. Under DNA, under herpes, we have herpes simplex 1, which is HSV1, HSV2, have varicella, we have cytomegalovirus, we have HHV 6, 7, EBV, and so on. All those are under herpes virus. Under picornavirus, we have poliovirus, hepatitis A, enterovirus, and rhinovirus. Let's take them one by one. Let's start with the herpes virus. Wait, you have a question? Thank you so much. Oh, does everyone want a break? No? يلا, not much left. تحمل شوي. Last lecture, تمام. طيب. Let's start with herpes virus. Herpes virus, we said, is an enveloped double-stranded DNA, which is Baltimore classification, Baltimore group. Wait, herpes virus. Enveloped double-stranded DNA. It has a large genome. And a lot of it, around 40% of this genome, is acquired from the host cell to protect this virus from destruction. So it's an enveloped double-stranded DNA, and it has a very huge genome, and it can persist as episomes, undergo latency, and then reactivate. We said which one undergoes latency in trigeminal ganglion, which virus? تمام. We said HSV1, herpes simplex 1, undergoes latency by forming episomes in trigeminal ganglion, and HSV1 is under herpes virus. Herpes virus can persist as episomes, they undergo latency, they get reactivated later on due to environmental factors, cytokines, hormones, and so on. And they're in high prevalence, they're common, basically, such as EBV or CMV. Now we're going to talk about each one of them and the infections they cause in the next slide, I believe. Yes. طيب. Those are the major herpes viruses we need to know. Starting with HSV1. HSV1, basic appearance would be cold sores and fever blisters. This, basically. Cold sores and fever blisters, and it's transmitted by kissing. So we said by direct contact. Either skin lesions or saliva will transmit HSV1. HSV2, on the other hand, is through sexual activity or intercourse, and it will cause genital lesions. So those lesions, as you can see here, are such as appear as vesicles, and this is caused by HSV2 in the genital region. Those can cause blisters, they can cause retinitis in the eyes, and they can undergo latency, and they're teratogenic. Remember we said teratogenic means it will affect the fetus development if the fetus acquires this infection. Moving on to the next group, which is varicella. Varicella zoster virus. We said it appears as chickenpox in kids, but it can undergo latency in the dorsal root ganglion and then gets reactivated when you're old enough, like 50 or 60. It will get reactivated as shingles in adults. But basically, chickenpox itself is self-limiting. In 2 weeks, you're done with the infection. You don't have the vesicles anymore. But if it gets reactivated, it can appear as this shingles. Basically, shingles will be following a certain dermatome because it undergoes latency in the dorsal root ganglion. When it gets affected, it will affect the dermatome of that specific nerve. So the shingles will affect this area. Yes. تمام. Now moving on to the next group, which is CMV, cytomegalovirus. Now, usually in normal cases, it's a controlled infection. In normal immunocompetent adults, it's usually controlled. But you have 2 cases where it's significant. So, first case is when it crosses the placenta in the first trimester. In other words, it can be teratogenic. So, when it crosses the placenta, it can give this appearance for the baby. They will have microcephaly, they will have calcifications in their brain, and it will cause basically problems with their development. This is the first case where CMV is significant. Second case is in immunocompromised adults. Who are immunocompromised basically? Transplantation or AIDS patients. In transplantation or AIDS patients, the previously controlled CMV infection can get reactivated and it will cause serious disease. So CMV can cause, for example, pneumonia. If you have transplant patient who suddenly had pneumonia, you should think about CMV. So CMV usually controlled, but in some cases it can be important. If it crosses the placenta, it can be teratogenic. And in immunocompromised adults, such as transplant or AIDS patients, it can get reactivated and cause disease. We also have EBV. EBV, we said, it will affect the B cells. It will cause uncontrolled replication of the B cells. Usually, it can cause infectious mononucleosis, otherwise known as mono, in like teenagers. This is basically a specific kind of infection that's not that serious, but it can cause very serious infections. It can cause cancers. We said EBV is oncogenic. So if the patient is immunosuppressed, it can cause B-cell lymphomas. It can cause B-cell lymphoma, Burkitt's lymphoma, nasopharyngeal carcinoma. So this appearance is for Burkitt's lymphoma. Basically, it's a type of cancer caused by EBV. EBV will cause a translocation in the chromosomes, and it will cause serious infection, cancer, which is Burkitt's lymphoma in this case. طيب. Now, we move on to picornaviruses. Now, from the name, picornavirus, you see it's an RNA virus. Pico means very small, very tiny. So you have picornavirus is the major family under RNA viruses. تمام? So it is a positive stranded single strand RNA. So here you have the RNA, a single stranded, it's a positive strand. And here you can see the envelope, the capsid, I mean. And it can be transmitted either by respiratory route or fecal-oral route, depending on the specific viruses under this family. Tamam? Let's take them bit by bit. Let's talk about the presentation of some specific viruses. So here you have hepatitis A, rhinovirus, and enteroviruses. Enteroviruses is basically a non-specific term, so it's not an actual classification. It's basically, it has some viruses under it. So hepatitis A, rhinovirus, we also have polio, Coxsackie, and echovirus under picornaviruses. Hepatitis A can cause self-limiting jaundice and can cause traveler's diarrhea. What is the infection, like the disease pattern? disease pattern is acute. So it's self-limiting jaundice due to the infection of the liver. It can cause traveler's diarrhea. This is for hepatitis A. Now for rhinovirus, we said it will affect the respiratory, it's a respiratory infection affecting the nasal epithelium. So this will cause common cold. Now we have enteroviruses, they can affect different organs. So it depends. For example, you have poliovirus. Poliovirus can affect the nervous system. So it's either can cause an acute infection or can cause paralysis. Coxsackie can cause something called hand-foot-mouth disease in kids. And you have echovirus. It isn't that significant, but echovirus stands for enteric human orphan virus. Yeah? Those are specific examples of picornaviruses, which is the biggest family under RNA viruses, positive strand ssRNA. Now, general classification by route of infection. You have respiratory infections, you have fecal-oral. Fecal-oral, we said rotavirus, epidemic in kids and daycare, and you have a vaccine for rotaviruses. So you have different ages, you give a vaccine for rotavirus to prevent this infection. Hepatitis A, you also have a vaccine for hepatitis A, and you have something called calciviruses. Under calciviruses, I know you have so many viruses, so many classifications, it gets very confusing, بس يعني تحملوا شوي. But under calciviruses, the main thing you need to remember about calcivirus, or in other words, norovirus or Norwalk-like virus, the presentation is vomiting and diarrhea. And when does this happen? It is common in gastroenteritis outbreaks, basically in cruise ships and military base, restaurants, and schools. So if you have a scenario where the patient was in a cruise ship and many people suddenly got vomiting diarrhea, it's usually norovirus. Norovirus is by the fecal-oral route and causes vomiting, diarrhea, gastroenteritis, outbreaks in cruise ship, military base, restaurants, and schools. By the respiratory routes, again, as we mentioned, you have influenza, you have RSV, which is respiratory syncytial virus, you have metapneumovirus, rhinovirus, coronavirus, all those are by the respiratory routes. Again, influenza. We said it undergoes many different mutations, and you have different species every season, and those different species will affect the vaccines. Yes. Wasn't Coxsackie equal? Okay, you have different classes under Coxsackie. You have different classes. Okay, now for influenza viruses. Influenza, you have mutations, and this would result in different species every season. And we said we need to monitor those to know how to formulate the vaccine. We are specifically concerned about H1N1 and H5N1 because those can cause major infections or major outbreaks in the population. And we talked about N and H and what they do. So H is for virion entry, N is for virion release. Tamam. And we said a major thing here happening is antigenic shift because influenza is a segmented RNA virus. So when two co-infect a cell, you can get strands from both strains, and this will cause a pandemic. It can also undergo antigenic drift, which is due to a mutation, but smaller scale. طيب, CNS viruses. CNS means it will affect CNS. عادي. So what viruses will affect the CNS? You have herpes virus. As we mentioned, HSV1 will be latent in the neurons. You have mumps. Mumps usually will affect, usual presentation will be a swelling here, but in males can affect the testes. It can also affect the CNS. You have enterovirus. As we mentioned, enterovirus, you have different viruses under it. One of them was poliovirus. Remember we said poliovirus can cause paralysis, so it affects the CNS. Rarely, Coxsackie virus can cause CNS infection too. HIV in advanced AIDS, so in advanced stages, HIV will also affect the microglial cells. Microglial cells are derived from macrophages in that lineage. So HIV can affect the microglial cells and they will cause dementia. Rubella. Rubella is also known as German measles. And remember we mentioned rubella as teratogen. So if it's passed from the mother to the baby in the first trimester through the placenta, it will cause basically problems with the fetus, with the maturation, the development and growth and so on. So it's a torch infection. Torch are the teratogenic infections. So next is rabies. Rabies is a zoonotic infection, as we mentioned. So zoonotic infection means it jumps species. So it can jump from the animals, from dog bite to the humans. And for that, you have passive vaccine. Passive vaccine means you do not give them the causative agent itself. Because usually in live vaccines, what you do in active vaccines, you give them the causative agent and you let the body do its thing. So you let the body activate the immune system, release the antibodies, and so on. And then it will make memory cells. So later on, when you get the infection, also you have memory cells and you're fighting the infection. Meanwhile, in passive vaccines, you give them directed antibodies. So you give them the antibodies to attack the infection right now. So if a patient for example, you got a kid who got bit by a dog, and now you're concerned about rabies. So you won't give them a live vaccine and wait for the body to try to fight the infection and make memory cells. No, you need direct action. You give them antibodies. You stop rabies from progressing because it can affect the CNS. So you give them antibodies right now. But passive vaccines will not cause long-term immunity. So you won't have memory cells formed because you're not giving them the causative agent itself and allowing the body to respond to that agent. No, you're giving them antibodies to fight the infection right now. You also have prions. Remember, we said prions in slow infection. So prions will be silent. They will slowly accumulate over years and then suddenly cause infection. This infection will affect the brain, CNS. Finally, we have arboviruses. Arboviruses, arthropods. Think arboviruses, arthropods. So think about insects. So arbovirus is vector-borne. So you have insects carrying this infection, carrying this virus and infecting the host cell or the humans. So here, arboviruses can be carried by arthropods, by ticks, mosquitoes, sandflies, and they will feed on your blood and then transmit the infection. So again, this is a zoonotic infection. It's jumping species. And it's mostly in hot tropical countries when human encroach tropical rainforests. Basically, what this means is they're usually just in animals, adi. They're not affecting anyone. Humans go to the tropical country, they go to the rainforests, and they get infected. So now it jumped species from the animals to the humans. And it's multi-system, so it will affect the endothelium, the CNS, the skin, and the muscles. And it can cause a hemorrhagic fever depending on the type of viruses. So some of them can cause encephalitis, so they cause infection in the brain. Some of them can cause hemorrhagic fever, and we're going to talk about the specific types in the coming slides. So under arboviruses, you have even more classification. Question? Does anyone have the slides? I will send the slides after the presentation, after the lecture. Tamam? Those tables are like copy-paste from your lecture, from Dr. Garman. Okay, you're welcome. Tamam. So here you have togavirus and flavivirus under arboviruses. So those are viruses that are transmitted by mosquitoes, arthropods, insects, basically. Togaviruses and flaviviruses, here you can see the specific locations where they cause infection. The ones underlined are basically major in Saudi Arabia. The others are less common in Saudi Arabia. They're more common in other places. So those are the ones we need to focus on. Zika virus, West Nile, dengue fever, yellow fever, but you need to know the rest. So for example, let's talk about Toga viruses. Toga virus family, you have alpha virus and drobivirus. So under alpha viruses are a big family. They have 28 members. Some of them are zoonotic and they're spread by mosquitoes. So what we have under alpha viruses, you can have the Western equine, Eastern equine, chikungunya, and all those are under Toga viruses. You also have ruby viruses, which only has rubella. Rubella, we said, is teratogenic. It can cause fetal abnormalities when transmitted from the mom to the baby in utero. So those are in Toga viruses. You also have Zika virus. Zika virus also can cause infection in the baby. It can cause microcephaly. It can cause many different things. And Zika virus is transmitted by insect bites. Under flaviviruses, you have West Nile virus, you have dengue, and you have yellow fever. Now, dengue fever is very important. You need to remember it is by Aedes mosquito, and It's very important, very high yield, and they usually get questions about this. But just know everything, and you don't ignore the rest. This is transmitted by Aedes mosquito. Usually have co-infection dengue with chikungunya. So both of them are transmitted by Aedes mosquito. You also have yellow fever. You have West Nile. Yellow fever will affect the liver, as we said. You have dengue fever. Dengue fever will, dengue will cause hemorrhagic fever. We're going to talk about hemorrhagic fever in a bit too. Those are the main things. We talked about Toga virus. Now, those are the things that cause encephalitis. So those are the viruses that will affect the brain. Under the alphavirus and flavivirus family. Under alphavirus, you have the Venezuelan encephalitis, Eastern equine, Western equine, all those, and Japanese encephalitis. Those all, till this one for alpha viruses, those all are under alpha viruses and they cause encephalitis. You also have West Nile and Japanese from flaviviruses under arboviruses causing encephalitis. So you need to remember those both. They usually don't ask about the rest, but you never know. Japanese encephalitis, West Nile virus, all those will cause encephalitis. Finally, those causing fever. You have dengue, Rift Valley fever, and yellow fever. Those are very important in the Middle East. Dengue, as I mentioned, will cause fever, hemorrhagic shock, rash, and all those. Dengue is very important. We also have Rift Valley fever, also fever and sometimes hemorrhage. Yellow fever, we said yellow fever infects the liver, so it will cause fever and hepatitis. And those are the main locations they usually found. I don't think you need to know the location in detail for this stage, but later on, sometimes the only hint that will help you answer the question is basically the location. So when they say the patient came from Africa, the patient came from Brazil, and so on, you will get a hint of how to solve the question. At your level, they're not going to ask in details about the location. Just know the main ones related to Saudi Arabia. And again, you have chikungunya, which is similar to dengue because they're both transmitted by Aedes mosquito. And you have Colorado tick fever. تمام. Finally, vaccination. As I mentioned, vaccination, you have a vaccination schedule that should be followed in the pediatrics, basically. So those are the approved vaccines for pediatrics, and adults have to take other vaccines, for example, influenza, which is seasonal, and specific vaccines in specific like conditions. Basically, let's start with those that are the approved vaccines. MMR, which is measles, mumps, rubella vaccine. You have polio vaccine. Polio can be either killed or live attenuated. Now, killed vaccine means it won't cause an infection. Live attenuated means you're taking the causative agent itself, and it may, in some cases, cause the infection. That's why I wrote beware here. So, if the patient is immunocompromised, it means that their immune system cannot handle a very strong infection. So, if the patient is immunocompromised, you need to think twice before giving a live vaccine. And in case of oral polio vaccine, you need to make sure that the patient is not immunocompromised. And also, if he has someone immunocompromised in the family, you do not give the OPV. Because if you give OPV, it can cause the infection. It can cause polio and will cause paralysis. Meanwhile, killed vaccine, which is the IPV intramuscular, it's safer, basically. Because it's a killed vaccine, not live attenuated. You also have yellow fever vaccine, hepatitis A and hepatitis B. Remember when I talked about hepatitis B, I said it's transmitted through blood, which means that you can get, if you get a prick by a needle or a syringe used by a patient who has hepatitis, you can get hepatitis yourself. So healthcare workers should get hepatitis B vaccine. Also have rotavirus. Rotavirus, again, can cause gastroenteritis in kids. So they usually have rotavirus vaccines. As you can see here in the vaccination schedule for the kids, you have multiple doses. HPV, human papillomavirus, because this will protect against cervical cancer. So HPV is usually given, in our countries, it's usually for females. In other countries, they usually give it also for males that are high risk. So it's usually given at age 11 to 14 years. And it's usually 2 doses, sometimes 3 doses, to protect against cervical cancer. And finally, influenza. Influenza is recommended for healthcare and for elderly because they're high risk. Healthcare because they can get it from the patients. Elderly because they have low immunity, so you need to give them the seasonal influenza. And seasonal influenza is usually given starting from 6 months of age and till elderly, and you give it seasonal every year. This brings us to the end of virology. Now we have the last set of questions for the last lecture. What is the virus that causes gastroenteritis in children? Rotavirus. Okay, what does rubella cause? Where did we mention rubella?
Is it tetragenic rubella?
Yes, rubella is the teratogenic one, which will cause infections in the fetus if it's passed transplacentally, which is why the mother needs to be protected against rubella. طيب, HSV1, what is the significant thing we mentioned about herpes simplex 1? Anything?
Lymphoma? Or
No.
Mm.
It's cold sores. Yes, and they undergo latency in the CNS. Yes, latency in trigeminal nerve, exactly. And yes, they will cause cold sores when reactivated. تمام. HIV, we said, can cause AIDS. So it's a chronic infection. And after a certain point, it will cause AIDS. It will affect the microglia cells and it will cause HIV dementia. HPV will cause cervical cancer. So you have a vaccine against HPV. Which of the following viruses is considered to be zoonotic? Have B, we have A. Yes. Okay, majority is right. So zoonotic means it jumps from species to species. We said dengue fever is, yes, by Aedes mosquito. So it can jump from the mosquito to the human. So it's a zoonotic infection. For those who answered A, rubella, we said it's transmitted from the mother to the fetus. You confuse between rubella and rabies. Rabies is the one that's transmitted from a dog bite to humans. That's rabies virus, not rubella. تمام? Again, the others are not zoonotic, so they're confined to humans. Last question. Okay, we got some news. Rest, what do you think? Do you agree? طيب, whoever said B, yes, you're correct. Basically, we said CMV, cytomegalovirus, is usually, yes, okay. We're not talking about rejection exactly, but transplant is the keyword, yes. Yes, so CMV is usually contained and a contained infection. So basically, it does not cause a major infection, major presentation. But what happens is, in some cases, we said CMV will be significant, either if it was a congenital infection or an immunocompromised. Immunocompromised if the patient has AIDS, if underwent transplant. So in transplant patients, the main thing you need to remember is CMV can cause several infections, including pneumonia, in immunocompromised patients. Hepatitis A, we said, is an acute infection, so it does not cause something major. It's just acute hepatitis, traveler's diarrhea, and then it resolves. CMV and transplant and congenital. HSV will have a latent infection, and then it will appear as cold sores if it's HSV1, genital lesions if it was HSV2. EBV can cause either infectious mononucleosis, Burkitt's lymphoma, which is the appearance, the huge thing that was here in this slide. Where is it? Yes. This is EBV. Burkitt's lymphoma, also nasopharyngeal carcinoma. CMV, we said in transplantation and AIDS patients, it will get reactivated and will be murdered. Ta-da! This brings us to the end of the crash course. Thank you so much for listening. If you have any questions, you can either ask right now or contact me here. And please scan the QR codes. Allah yafikum. Just thank you so much. I'm always here to help and answer